Today I would like to give lecture on Introduction to Immunology and Humoral and cell-mediated immunity. Okay, so what is the immunology? Immunology is a branch of science that particularly deals with the immunity. Like immunity is nothing but inborn or acquired resistance of living organisms to infectious microorganisms. So that is known as immunity. So it deals with the study of processes by which the body defends itself from the invasion and attack of various types of foreign microorganisms like pathogenic microorganisms. So the overall study of resistant power or defense mechanism known as immunity. So types of immunity. Immunity is basically two types. One is known as innate immunity, another one is known as adaptive immunity. So what is the meaning of innate immunity? Innate immunity means from by birth the individual has resistance to the pathogens, that is known as innate immunity. In that like it is a kind of non-specific immunity and it provides the first line of defense and as well as second line of defense to the infectious microorganisms. So it is present inside the body before the infection, and it shows a set of resistance mechanisms towards the pathogenic microorganisms. So for example, if you take macrophages and neutrophils and skin on the human skin, these will all act as a mechanical barriers or resistant power containing components in the innate immunity. So what are the properties of this innate immunity? Like it shows rapid reaction and it do not have any immunological memory kind of response, and also that immunity, mainly this innate immunity depends on germline encoded receptors. So those receptors are involved in the recognition of these pathogenic microorganisms. And this innate immunity also, it does not show any clonal expansion of cells towards the antigen, and it acts quickly towards the elimination of the pathogens, like within 4 hours it shows the reaction. And also it can differentiate the host cells and as well as pathogenic cells, and moreover it is more common to the all pathogens. So that is known as innate immunity. So first this Metchnikoff, he first worked on this innate immune response, and also he invented the phagocytosis mechanism, and he shared the Nobel Prize with Paul Ehrlich in 1908, and also he is called as father of natural immunity. So another one is that adaptive immune response or adaptive immunity. So during in his lifespan, that individual acquires or that individual exposes various types of diseases during his lifespan. By exposure of that disease, that individual will get the immunity. Such type of immunity known as adaptive immunity or specific immunity or acquired immunity. So these resistance power or this defense mechanism, mainly it is adapted or it is acquired from exposure of the pathogenic microorganisms. So this adaptive immunity has specific characteristics like antigenic specificity, diversity and immunological memory, and also it has self and non-self recognition of the particular pathogenic cells and as well as non-pathogenic cells. So these are the main 4 characteristics of adaptive immunity. So these adaptive immunity or acquired immunity initiated by recognition of antigen by specific lymphocytes or specific cells called T cells and B cells, and activation of these specific lymphocytes undergo into the proliferation and as well as differentiate into effector cells, and these effector cells eliminate the antigen, and after elimination of that antigen or pathogen, it will come back to the homeostasis, and in that process it also develop the memory cells. So these memory cells involved in the second exposure of the same antigen in the second immune response, it can rapidly divide into plasma cells and it produces the antibodies. So these are some of the characteristics related to the acquired immunity. So Paul Ehrlich is contributed most of this work in the adaptive immunity, and he shared the Nobel Prize with Mechnikov in 1908, and is also called father of modern immunology. So this acquired immunity or this adaptive immunity mainly involves a combination of two mechanisms known as humoral immune response and as well as cell-mediated immune response. One is humoral immune response or humoral immunity or antibody mediated immunity. Another one is cell-mediated immunity or cell-mediated immune response. So before going into these mechanism, first we will see what are the components required to deliver these functions in the humoral immunity and as well as cell-mediated immunity. First we will go into the cells in the immune system. So some of the particular cells, they are involved in the getting up the resistance power. So like these are originated from the bone marrow, from the stem cells like hematopoietic stem cells, and they are basically that stem cells convert into two lineages. One is called myeloid lineage, another one is called one is myeloid lineage, another one is called lymphoid lineage. In the myeloid lineage, you will get some of the megakaryocytes and erythrocytes, mast cells and myeloblasts. At the end, this myeloblast, it will give the basophils, neutrophils and eosinophils and monocytes. At the end, these monocytes, when they are present in the tissue, they used to call as macrophages. Same way, in the lymphoid lineage, there are two types of cells. One is big lymphocytes or large granular lymphocytes. Another one is known as small granular lymphocytes. So in that, so these large lymphocytes, they are called as natural killer cells, and these small lymphocytes, again they are divided into T lymphocytes and as well as B lymphocytes. So where they are exactly involved? So these basically they are involved in either innate immunity or adaptive immunity. Like if you take the myeloid cells, basically they are involved in neutrophils, like getting up these neutrophils and eosinophils and basophils are monocytes, macrophages and dendritic cells. They are involved in the non-specific innate immune response or non-specific immunity. Whereas these lymphoid cells are lymphoid cells of the natural killer cells. They are involved in the non-specific immunity. Means these are intermediate between the innate immunity and as well as acquired immunity. So next, these lymphoid cells like T cells and B cells, they are involved in the humoral immunity and as well as cell-mediated immunity. So when you look into the lymphocytes, like lymphocytes are two, based on their function, there are two types, T lymphocytes and as well as B lymphocytes. T lymphocytes, again based on the function, they are divided into different types like T helper cells and T cytotoxic cells and as well as natural killer cells. So these T helper cells, again they are divided into, they undergo into the differentiation. After activation, they will be converted into T helper naive cells, and these T helper naive cells can be differentiated into T helper 1 and T helper 2, and as well as these T helper cells undergo into the T regulated cells, and these T regulator cells are two types, natural T regulator cells and as well as adaptive T regulator cells. These adaptive T regulator cells also called Th3 cells. So next, when you look at the these B lymphocytes are two types, B1 and B2, and these B1 and B2, these two are mainly involved in the production of antibodies. They are again, after activation of these B1 and B2, they undergo into the differentiation. They will form the plasma cells, and plasma cells divided into memory cells and as well as antibody producing cells. So these are the kind of lymphocytes they are involved in the specifically they are involved in the acquired immunity. So if you look at this structure of these T helper cells like T helper cell and as well as cytotoxic cell, it has big nucleus and it has less amount of cytoplasm, and as well as on the surface of this cell it has various number of receptors or glycoprotein molecules. So basically you need to remember the T cell receptor called TCR, T cell receptor CD3 and as well as CD4 and as well as CD8 and Thi1 and CD7 like that and CD28. So if you specifically see the T cell receptor, they have specific kind of receptor that is known as CD4. So that's why they used to call CD4 positive cells. And if you look at the cytotoxic cells, they do have CD8 as a specific receptor along with the TCR and CD3 and CD28 and Thi1. So these cells they used to call as CD8 positive cells. So basically these are all are involved in the receptor specificity and adhesion type, and also they are involved in the cell signaling process. So next other cells called antigen presenting cells. So basically these are like what they will do, they will go around the body and they will check the any pathogen is there or any antigen is there, they will capture it, and after that they will undergo into the processes like phagocytosis processes. After phagocytosis process they will digest into small species. After digestion they will express it on the surface of that antigen presenting cells with the help of MHC molecules. So after this they will go and present to the particular type of cells. These antigen presenting cells, they will present to these antigen or pathogen to the T lymphocytes or B lymphocytes. So if you take the example like the best antigen presenting cells are macrophages, dendritic cells and B lymphocytes and neutrophils and eosinophils. So these are all are involved in the capturing of microorganisms or antigens and process it and present to the T cells. So next another one is that major histocompatibility complex. Major histocompatibility complex. So these major histocompatibility complex has two types. Basically it has two types like class 1 MSC molecule and class 2 MSC molecules. These class 1 MSC molecules mainly it is expressed in all the nucleated cells. Class 2 MSC molecule it is present in the antigen presenting cells. So these are involved in the presentation of the antigen to the T lymphocytes. So what are the antigens? So antigens are nothing but the part of the microorganism. When that enters into the body, that part of the microorganism can act as an antigen. Like these are most of the these are like products or most microorganism products of the proteins or large polysaccharides from the microbes. And microbes also they contain like capsule, cell wall, toxin or viral capsids or flagella. These are all act as a antigens. Like non-microbe antigens like pollen grains or egg white, red blood cell membrane surface molecule, serum proteins. These are all are act as a antigens. So like in the microorganism they do have like lipids and as well as nucleic acids. These can also act as a antigens. This antigen if the molecule of that microbial product or microbial constituent can act as an antigen, it should have molecular weight of above 10,000 Dalton. Above 10,000 Daltons. So these molecules like basically antigens are nothing but they are part of the microorganism, they are part of the pathogen, they are constituents of the pathogen. Example you can take cell wall or capsule like that. So come back to our original topic, humoral immune response and cell-mediated immune response. So what is the humoral immune response? Humoral immune response is nothing but, so when this pathogen enters into the human body, that stimulates the production of various types of immunological cells like lymphocytes. After activation of these lymphocytes, at the end they produces the antibodies. So by exposure of microorganisms stimulates the production of antibodies. That process or that response known as humoral response and also called humoral immunity or antibody mediated immunity. So in that basically we have T cells and as well as T helper cells. So these T helper cells recognize the antigen presenting cells, then they undergo into the differentiation. After that they stimulates the B cells, and after that these B cells undergo into the differentiation, they will form the plasma cells, and these plasma cells produces the antibodies. So these antibodies mainly they are present in the blood or blood plasma, lymph and mucus, so like that. So they are only present in the extracellular fluids. So these where they will go after production of these antibodies, and they will go at the site of infection, and they interact with that pathogen like bacteria or bacterial toxins or viruses, and they eliminate that pathogens they before enters into the cells. So here look at this mechanism of humoral and immunity and cell-mediated immunity. So this is the so skin. It is a human skin upper layer portion. So this skin act as a mechanical barrier for prevention of microorganisms, prevention of entry of microorganism into the inside the body. So this basically this skin also it produces sebum and as well as sweat. So these sweat and sebum they contain some of the antibacterial substances, and it has low amount of pH. Due to that, it eliminates the pathogens. Basically, it will prevent the attachment of pathogens. And this skin also it harbors the normal flora. So this normal flora also prevents the attachment of microorganisms. So that's why this type of resistant power called innate immune response or innate immunity means by birth that individual has some resistance power. Naturally that individual has some resistance power or some defensive mechanism in the form of skin. So that is called innate immunity. Some microorganisms they specifically escape this skin and they enters into the connective tissues of that skin cells. Between the tissues you have intercellular space. Suppose you had one injury, because of this cut, this cut act as an entry site for the microorganisms. So these microorganisms enters into your intercellular space in the skin cells. So this basically what this microorganism will do, it will roam around here and there, and it will see where that microorganism want to hide inside the particular cells. So it will see here, and it will move here and there, and after that it will go into the cells, or it will still remain inside that intercellular space. So this intercellular space also it is connected with lymph or blood or blood vessels. So these blood vessels are all connected into all the intercellular spaces. So in that blood or in that lymph you will find the macrophages or antigen presenting cells, dendritic cells or neutrophils like that. So this is the example for the antigen presenting cells, example macrophage. So these macrophages mainly they are present in the lymph and as well as in the blood. So these macrophages will go around the intercellular space and they will check any unwanted microorganism is there, or any foreign microorganism is there, or any pathogenic microorganism is there. See, they will check inside the intercellular space. After that, if it is finds, if that pathogen finds, it will capture with this pseudopodia and it will engulf it. It will undergo into the phagosome formation, and that phagosome fuse with the phagolysosome, and after that it will undergo into the digestion process. If suppose if that microorganism do not want to stay in the interstellar space, so where it will go? It will enters into the cells. It will enters into the inside the cell, particularly intracellular pathogens. Intracellular pathogens, they do not reside. They do not reside in the interstellar space. They will go inside the cells. So if suppose that pathogen want to go inside the cell, then if that pathogen want to go inside the cell, it cannot be recognized by macrophages, because macrophages they are present only in the blood and lymph. It cannot see the particular cells. It cannot go and see inside the cell what is going on. So that's why some of the intracellular pathogens they will hide inside the cells. So here, so these macrophage captured the captured the one pathogen. So after capturing it processed it, then after processing it produces some of the interleukin molecules called as cytokines. They produces the cytokines. So means, hey, I got something inside my body, so I want to expose that one in the form of message or in the form of chemical messenger. So they are called as cytokines. So these cytokines are released from that antigen presenting cells. So that one of the cytokine known as interleukin 12. Interleukin 12. This interleukin 12 specifically stimulates the T helper cells in the T helper cells. Actually, these T helper cells are they are in the form of naive T cells, naive T helper cells. Naive means they do not know anything. They do not know what they want to do in the future. When this interleukin 12 binds to its receptor on the naive helper T cell, after binding up this interleukin 12, it will undergo into the activation process. In the activation process, these naive T helper cell will be converted into Th1 cell, T helper cell 1 cell, T helper 1 cell. So this T helper 1 cell specifically again it will stimulates and it produces some of the interleukins called interleukin 12 and interleukin 12. So these interleukin 12 itself act on its Th1 in the form of autocrine signaling, and as well as this interleukin 2 can also stimulate the activation of TC cells. These are called T cytotoxic cells or CD8 positive cells. CD8 positive cells. So these CD8 positive or cytotoxic T cells, these T cells are basically they are involved in the cell-mediated immune response or cell-mediated immunity. Okay, once again I will repeat it. So first this pathogen captured by the antigen presenting cell, so then it stimulates the activation of naive T cells, particularly naive T helper cells. Then that naive T cells, maybe they will be converted into Th1 cells. In turn, these Th1 cells again it produces the interleukin 2. These interleukin 2 again it produces the it activates the T cytotoxic T cells. So this is one kind of mechanism. Look at another one. In turn, this T helper 1 cell also it can also produces the interferon gamma. In turn, this interferon gamma also stimulates the more amount of production of antigen presenting cells in the form of macrophages. So this is one kind of response from the pathogen. So another response from the same pathogen, like that unknown cell, it produces the interleukin 4. Unknown cell, we do not know till now what is the cell. It produces the interleukin 4. By the action of this interleukin 4, that naive T helper cell will be converted into Th2 cell. Th2 cell, T helper 2 cell. So these T helper 2 cell in turn it produces the interleukin 4 and as well as interleukin 5. Interleukin 4 and as well as interleukin 5. These interleukin 4 and interleukin 5, they will activate the B cells. They will activate the B cells. After activation of these B cells undergo into the differentiation, they will form the plasma cells. So these plasma cells, it produces the antibodies. So these antibodies are specific to that particular type of antigen which is entered into the body. So these are the two, these process like interleukin 4, Th2 helper cell and as well as B cell and plasma cell and production of antibodies. This mechanism or this process known as humoral immunity. Humoral immunity. Okay, first we will see what is the humoral immunity. So this is a pathogen. It enters into the it is captured by the antigen presenting cells in the form of macrophage. It will be digested it, After digestion, that peptides or that small molecules will be presented to the lymphocytes in the form of T helper cells with the help of MHC2. MHC means major histocompatibility complex, major histocompatibility complex. So once after processing with the help of this MHC with that peptide, it will be present on the surface of that APC. So now this MHC with along with that peptide interact with the TH2 cells, TH2 cells, that activated T helper 2 cells. So after this interaction, so in the TH2 cells they do have TCR, T cell receptor. So this T cell receptor interact with the that antigen or peptide processed from the antigen presenting cell. After that, this interaction again it is strengthened by the formation of interaction between the MHC2 and as well as CD4 receptor. Formation of these kind of interactions, then this T helper 2 cell produces the interleukin 4 and as well as interleukin 5. So these interleukin 4 and interleukin 5 and they binds to the receptor on the B cells, and B cells are undergoing to the activation and as well as differentiation. Then after that they will be converted into plasma cell and as well as memory cells. So these plasma cells at the end they produce the antibodies. So these antibodies actually they are produced in the lymph nodes and they will travel towards the site of infection with the help of lymph. So what is the function of the memory cells? So memory cells basically they remain calm a long period of time. When the same type of antigen exposes in the secondary immune response, then rapidly these memory cells will be converted into plasma cells. They produce the antibodies. So this is called humoral immunity. Then when we will see the cell-mediated immune response, cell-mediated immune response. So the immunity which is getting mainly from the cells, from the lymphocytes. So such type of immunity known as cell-mediated immunity or cell-mediated immune response. So here we do have like again here we have T lymphocytes in the form of T helper cells. So they recognize the foreign antigen by means of surface receptors, and these T lymphocytes attack and destroy the foreign antigen infected cell directly or through release of some of the soluble mediators. So this is the T cytotoxic T cell. So it has the TCR. So then this cytotoxic T cell it interact with the virus infected cell like intracellular pathogens, or like example you can take mycobacterium, tuberculosis, or some of the viruses or some of the intracellular obligate intracellular parasites. They will all act as a endogenous antigens. So this T cytotoxic C cell it will interact with the that infected virus infected cell with the help of T cell receptor, and these virus infected cell they also expresses the low amount of MHC1. These MHC1 interact with the TCR along with the processed antigen. Then after this interaction also strengthened by the formation of interaction between CD8 and as well as MHC1 molecule leads to production of some of the perforins and gran enzymes in the T cells. So these perforins and gran enzymes